Hello, everybody. How are you? Here we have reached to the fourth evidence number four. The fourth evidence is, as you observe, evidence from comparative biochemistry. Biochemistry, of course. We shall examine biochemistry, biomolecules, biochemicals in a comparative manner. The more similar in biochemistry two organisms are sharing, the more closely related in terms of evolutionary history. This is a rule for comparative biochemistry. We are comparing different chemicals like DNA and proteins, including hemoglobin, of course, organisms that share very similar molecules or biological molecule, biochemical pathways. This also includes, of course, pathways like glycolysis. Glycolysis is common for all. Why? Of course, the first evolved. Therefore, biochemical pathways, energy production pathway, or protein synthesis pathway, or even gene expression is biochemical pathway itself. Therefore, in biochemistry, we compare biomolecules or molecules of life plus biochemical pathways that are closely related in evolutionary. Therefore, organisms that share very similar molecules or very similar biochemical pathways do have close relation in terms of their evolutionary history. Chemicals used for such comparison include DNA and proteins and amino acid squares in a polypeptide, of course, squares of amino acids and amino acid content, the squares, and also the protein profile, the protein shape, and the three-dimensional structure. Uh, Species that have the most similarity in DNAs and proteins are believed to be very much close to each other in terms of their evolutionary history. And the opposite is also true. Species that have the less similarity in their DNA or proteins are believed to be distantly related. They are far away. And example, comparison of DNA sequence of humans and chimpanzee shows 98% similarity, which confirms that chimpanzees are the most closely related primate to humans. 98% of the DNA of chimpanzee and DNA of humans similar means the most closely related to humans out of the primates, the apes, the great apes. We call them the great apes. Like the great apes are the gorilla, the orangutan, the gibbons, chimpanzees. From all these great apes, chimpanzee is the nearest to humans. You can see this from the 98% similarity in the DNA. If this evolutionary tree, also known as phylogenetic tree, shows some animals based on difference in their DNA. You can see the difference. You see, therefore, our DNA, if it's 100%, 99.01% similar with chimpanzee and other figure. Of course, some literature declare that 98% of the DNA of chimpanzee is similar to that of humans. But here, 99.01. I think this must be different research. The other next to chimpanzee, gorilla, 98.90%, and the African monkey, 96.66% DNA similarity, of course. This is similarity. Hemoglobin, the other molecule that we use, scientists are using to compare as evidence for evolution is hemoglobin. Hemoglobin in those animals which contain hemoglobin, hemoglobin molecule is similar in all animals that possess it, but there are differences. When we say they are similar, still there are differences. There are differences in those that are distantly related. Lampreys, for example, lampreys a primitive fish-like animal has hemoglobin with only one polypeptide. Otherwise, all the other animals which possess hemoglobin, most of them do have hemoglobin molecule made from four polypeptides. Four polypeptides, huge molecule, of course. The four polypeptides join together with prosthetic group by prosthetic group, non-protein part that contains iron also. They are joined together to produce that four-dimensional, I mean the quaternary structure protein, that is hemoglobin. Therefore, four polypeptides, of course, two polypeptides are similar, known as alpha. The others are beta in adults, of course. There is also another polypeptide which is known as gamma. I will show you this. When it comes to infantes, the composition is different. Most other animals possess hemoglobin with four polypeptides, but the polypeptides are still different in different animals. How are the polypeptides different? The sequence of amino acids. Even the number of amino acids may also differ in some. Sequence of amino acids, how amino acids are arranged. For instance, they show differences in amino acid sequence of the alpha chains of hemoglobin of humans and several other animals. The table in the next slide shows comparison of the polypeptide chains of hemoglobin in humans and three other primates. This is the table, of course, that shows you. This is a table. This table shows you hemoglobin difference between the polypeptide chains of hemoglobin in four primate species. These are human, chimpanzees, gorilla, and gibbon. And it shows the alpha hemoglobin with 141 amino acids, chain of amino acids. Polypeptide chain beta hemoglobin, it contains 146 amino acid sequences in all these primates, and gamma hemoglobin, as the amino acid sequence is not known, of course. Then when it comes to humans, you see the plus sign is, of course, hemoglobin. Plus sign is, of course, hemoglobin, as I've told you, is composed of four polypeptide chains, the alpha, beta, gamma type, four. In adults, two beta, two gamma. Plus indicates no difference in amino acid sequence from that of human, and the numbers indicate the differences in the amino acid sequence. You see now, when you go to human, compare alpha hemoglobin 141 amino acid chains, and chimpanzee, no difference with chimpanzee, and in alpha hemoglobin. In beta hemoglobin, still the human beta hemoglobin has also no difference. Chimpanzees, still no difference. Gamma hemoglobin also no difference. The human and the chimpanzee polypeptide, no difference. You see, this indicates how much we are similar with polypeptide chains of the alpha, beta, and gamma. When it comes to gorilla, the human and the gorilla, there is one amino acid sequence difference. Between human and gibbon, three amino acid sequence vary. And in case of beta hemoglobin, between human and gorilla, there is still one difference, and between human and gibbons, three differences. You see, gibbons are much distant as compared to chimpanzee and gorilla. In case of gamma hemoglobins, the same with gorilla, just one amino acid difference, and with gibbons, two amino acids differ. You see, therefore this is comparative anatomy. And comparative biochemistry. Comparative biochemistry, we compare different biological molecules. If you go to plants, you compare the different types of chlorophyll molecules, chlorophyll A and B and the carotenoids. And if you go to animals, you compare the different types of proteins, enzymes, including the oxygen carrying molecule, that is hemoglobin, if the animal do have that hemoglobin, of course. Therefore, you see, this is evidence. If you ask this question, why the hemoglobin of chimpanzee and human similar this much? If you ask a religious personality, we don't worry. God made that to happen. It's the will of God. Don't worry. Let me tell you, don't worry. But when you come to science in evolution, this is an indication that humans and chimpanzees share very near evolutionary history, very close resemblance in terms of evolution. The other is here. The diagram shows you the evolutionary relationship of some animals shown by differences in their hemoglobin. You can go to this lamprey. I have told you lamprey. Lamprey is much distant from humans. Lamprey, of course, lamprey hemoglobin is composed of only one polypeptide. One polypeptide. Therefore, our hemoglobin is much similar to macaque. It's a monkey, macaque monkey, then dog comes, then bird, then frog. And lamprey is much distant from both humans and all the others, you see. And therefore, by comparing the biochemistry of different animals or plants, we can have such a tree, evolutionary tree, phylogenetic tree. The other evidence is evidence from paleontology. Paleontology. What's paleontology? Fossil record, the study of fossils. And this is the strongest, of course, the strongest evidence of all. Paleontology, or paleontology written in different way, of course, is the study of fossils. And fossils provide the strongest evidence for evolution. And what are fossils? First, let us see what fossil means in this context. And fossils are formed when certain remains of organisms get embedded in the soil or water and are preserved for many hundreds of years. Remains of any remain of an organism, an animal or plant, fossil. Uh, they appear as skeleton or as footprint, even footprint is considered as fossil. Molds, molded, molded part of an animal, molds, intact structures, especially intact structure with no part missing. You may get such fossils if the animal is accidentally buried under the ice in the polar areas. Intact structures or intact animal, you can call it. By studying fossils, we are able to establish similarities between organisms in the present and their ancestors in the past. You can compare it with those organisms that are living now, and also we may trace their ancestry back by fossils, rating these fossils, discovered fossils. This can be many. There can be many similarities that prove the common origin between different closely related animals, and the differences can be studied to establish how they differ, and now how they differ now, and why they differed, what evolutionary line they followed. You see, many similarities you may get that prove the common origins between different closely related animals, and the differences can be studied to establish how they differ now and why. Fossils are very important evidences to prove the theory of evolution and common ancestry, and we can group fossils into two categories. The first category, category one, fossils, the remains of dead animals or plants, or the imprint left from the remains, and include this one, category one, bones, teeth, skin impression, hair, the hardened shell of an ancient invertebrate such as the trilobite or ammonite. These are fossils discovered um, fossils of invertebrates discovered and are available. And an impression of an animal or plant, even if the actual parts are missing, tiny impressions. And category two, uh, fossils are something that was made by the animal while it was living and that it has hardened into stone since then. And these are called trace fossils and include, these are of course category two fossils are just found in sedimentary rock, demerged into rock. You see, something that is part of the animal or made by the animal while it was living and that it has hardened into stone since then. These are called trace fossils and include footprints are also categorized under category 2 fossils. Burrows, holes in which the animal used to live, they provide evidence, little evidence, trace evidence. The other is coprolite. Coprolite means the faces, animal faces. The waste material is also used as category 2 fossil, of course. Type 1 fossils can be the actual organisms or part of an organism, like a piece of bone or hair or feather, as it is as it actually was, just part of the organism, the whole organism. It could be, of course, type 1, category 1. For example, the spider shown in the deer and the fig has been trapped completely unchanged inside the amber plant resin, plant material, complex polysaccharide that plants secrete, accidentally uh, dropped on a living spider millions of years ago and remained just like this. Amber resists decomposition. That's why the spider being trapped inside the amber resin has been discovered by archaeologists. Still, it's available as evidence. The soft part of the body has been lost and the exoskeleton completely preserved inside this amber, of course, as you observe. And recently, of course, scientists also obtained the whole elephant that has been extinct many years ago. They call it Mamo's elephant. While digging in the under ice, buried under ice, they dug out the intact, you see, just natural refrigerator preserved the whole body from decomposition and from being eaten by other animals. And they dug out, and now, in fact, they are trying to bring that Mamo's elephant back by cloning. They, they will take cell from DNA, then they are trying to bring this animal back into life by cloning, using cloning technology.